Hello, this is Angel. Let us study mitosis together. Alright, so let's recall a little. Chromosomes are thread-like structures, as you can see. These are chromosomes, and chromosomes are thread-like structures that are made up of molecules of protein DNA, responsible for transmission of traits from one generation to the next. Okay, so it can be a trait how tall you can be, how small you can be, or dwarf you can be, or what's the skin color, etc., etc. So your chromosomes are thread-like and they carry protein DNA. Alright, so a haploid cell represented by symbol N contains only one set of chromosome, and gametes are haploid, right? So gametes are haploid, and they are only represented by symbol N because they carry one set of chromosome. So haploid cells are cells that contain a single set of chromosomes or half the number of chromosomes found in diploid cells. In humans, for example, somatic cells, normal body cells, soma means body, contain 46 chromosomes, while egg and sperm cells, gametes, contain 23 chromosomes. Haploid cells are produced through a type of cell division called meiosis, which reduces the number of chromosomes in a parent cell by half. When a sperm fertilizes an egg, the resulting zygote cells has two sets of chromosomes, one from each parent. A diploid cell is a cell that contains two complete sets of chromosomes, meaning it has two copies of each chromosomes, one inherited from each parent. Most cells in the human body are diploid, with 23 pairs of chromosomes, totaling 46 chromosomes in each cell, right? So diploid means double set, referring to the presence of two sets of chromosomes in a cell. In a diploid organism, one set of chromosomes comes from the mother and the other set comes from the father. Most cells in human body called somatic cells are diploid. In contrast, sex cells, gametes like sperm and egg, are haploid, meaning they only have one set of chromosomes. So a haploid have one set of chromosomes, and diploid has double set of chromosomes. Remember, it's not double, it is double set of chromosomes, okay? So this is haploid, one copy of each chromosome, 3 non-homologous chromosomes, and diploid is two copies of each chromosome, 3 pairs of homologous chromosomes of maternal and paternal origin, right? So what is the difference between haploid versus diploid, right? Refer to one set of chromosome is N, so this is N. In human, N equals to 23 chromosomes. Only gametes, sperm, and ova are haploid, so this is a sperm and ova, they're haploid. Diploid is 2N, refer to pairs of chromosomes, 2N, one from each parent. So one from mom, one from dad, and it's a set of chromosomes versus just one chromosome. As you can see, it's one here, right? In human 2N, 46 chromosomes in total. Only somatic cells are diploid. Or here, only gametes, sperm, and ova are haploid, right? Okay. Cell parts involved in cell division. Centrioles play a very important role. They're a pair of structures in animal cell involved in cell division, right? So these are centrioles. They're right here, okay? Cell parts involved in cell divisions are centromeres, right? It's a region of chromosome kinetochore where two sister chromatids are joined, where spindle microtubules attach to the chromosome during mitosis and meiosis. So this particular here is centromere, holds two chromatids together, and it's holding two chromatids together. This whole thing is chromosome, right? Gene, right here, these are genes. Segment of DNA that codes for a trait. These are chromatids, identical copies, right? The chromatids are identical copies. Centromere, this is telocentric, it's in the end. This is acrocentric, it's right here. This is submetacentric, it's closer to the middle, but it's not that close, right? So this is metacentric. This is the P arm, and this is the Q arm. P, Q, right? The top area is P, the lower bottom is Q. Cell parts involved in cell division. Cytoskeleton, the network of fine fibers that provide structural support for a eukaryotic cell. The thickest of the three main kinds of fibers making up the cytoskeleton is called microtubules, right? So this is particularly Cytoskeleton, the thickest part, the thickest of the three main kinds of fibers make up the cytoskeleton, and they're called as microtubules, right? So ribosome, cell organelles where protein molecules are synthesized. This is a rough endoplasmic reticulum. The reason why it's rough is because it has ribosomes on it. These are the free ribosomes, right? As you can see, these are the free ribosomes. Ribosomes are the protein factories. Ribosomes are associated with rough ER, right? So right here, they're protein factories. Alright, so nucleolus, structure in the nucleus where ribosomes are made. So this is the nucleolus, and these are ribosomes attached to the nucleolus. Right? So ribosomes are made in nucleolus. Alright, so what are the different steps of mitosis? Interphase. This is prophase, right? This is, interphase is like the starting, inception. In reality, it starts at PMAT, prophase. Here it shows prometaphase, but usually, whenever we discuss, it's usually prophase, metaphase, anaphase, and telophase, and that's it. It's usually PMAT or IPMAT, okay? So prophase, prometaphase, as you can see, this is metaphase, meta, plate. They're all arranged in a plate. Anaphase right here, this is anaphase, and this is telophase, two daughter cells, okay? Mitosis takes place in our body cells called soma, so it can be in brain, it can be in blood, heart, muscle, pancreas, eggs, sperm, kidneys, liver, bones, etc. Mitosis is responsible for increasing weight and height of the baby girl as she grew and developed into a teenager and then the old age, right? So it is because of mitosis. Process responsible for healing of the wound when the body part of the body is accidentally cut or scratched, right? So this is inflamed phase, day 1-3, right? So if you're a podiatrist, the reason why you'll see wound healing is because of mitosis. Collagen building phase is day 6-20. Remodel skin phase is 3 to 24 months. Responsible for regeneration of the body parts. This is a gecko, right? Or a chameleon. Chameleon can actually regenerate their body parts. So if you see chameleon with a broken tail, which happens very often because it's very slender, it grows back on its own, right? Or even if you actually see a broken tail, the tail will be wagging on its own, because it has the cells that keeps it alive. Mitosis is a process in which a eukaryotic cell nucleus splits in two, followed by the division of the parent cells into two daughter cells. The word mitosis means threads, and it refers to the thread-like appearance of chromosomes as the cell prepares to divide, right? So this is a spindle pole right here. Replicated chromosomes with sister chromatids. These are sister chromatids. And that is the little blue point is called as kinetochore, right? This is aster microtubules, right? It's a part of cytoskeleton. Kinetochore microtubules. It is also a part of cytoskeleton. Interpolar mitotubules, in between polars. The 5 stages of mitosis: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Usually they also talk about cytokinesis after. Here we will talk about it together with telophase. Alright, so how to remember all the phases of mitosis? IPMAT, okay? Interphase means it means between. Prophase means early, right? Prokaryote, pro is early, right? And eukaryote is like after nucleus, right? Pro is before nucleus, so pro is early. Meta is in the middle, right? Metaphase, right? It's in the middle. Anaphase, ana is going away, right? Ana. Anytime you hear ana, it's going away. Telophase, telo equals to end, right? Interphase, prophase, metaphase, anaphase, and telophase. This is under the microscope, right? So as you can see, this is the interphase, this is the prophase, this is the metaphase. Anaphase, they're dividing into two. Metaphase, they're coming in the middle. Pro is early, right? So this is interphase, in middle, right? Telophase. Telophase is 2 daughter cells. As you can see, there are 2 daughter cells formed. Interphase is a phase of the cell cycle when a cell grows, replicates its DNA, and prepares for cell division. Interphase as G1 phase, the cell performs its normal biochemical functions and begins synthesizing the molecules it will need to become two separate cells. So, you need molecules, so it starts in G1 phase, synthesizing the molecules. It's like you're putting gas in your car. Why? Because you have a long way to go, right? So S phase, the cell replicates its DNA, right? G2 phase, the cell prepares for mitosis, right? Interphase was previously called the resting phase, right? But the cell in interphase is not simply dormant, it's also known as the daily living or the metabolic phase of the cell, right? After interphase, the cell undergoes mitosis and completes its division. It's like you have a race car and the car is actually going for a race, right? So what you're doing is you're parking your car before you start, competing against another race cars, right? So that's what this is like a preparatory phase, right? This is interphase. As you can see, this is S phase, DNA synthesis. This is where the molecules get synthesized. This is G2, the cell growth, all right? And this is the mitotic phase, the mitosis, the cytokinesis. This is cell growth again, and the circle keeps on repeating. And after mitotic phase, there's cytokinase. The telophase is where you form two daughter cells. Prophase. Prophase is the first stage of mitosis. A type of cell division is characterized by several events, including chromatin condensation. The DNA and protein complex in the nucleus called chromatin condenses and coils into visible chromosomes. Nuclear envelope breakdown. The nuclear envelope breaks down, right? So the first thing happens is chromatin condensation. DNA protein complex in the nucleus called chromatin condenses and coils into visible chromosomes. Here I am, I'm going to replicate, so I might as well just be visible, right? So this is like an early preparation, right? The nuclear envelope breakdown, right? So nuclear envelope breaks down. You're coming out of something, you want to make sure that you're breaking down your envelope. That's what it is. You're declaring yourself that, hey, I'm a chromosome, this is what I'm going to do, so be ready. Nucleolus disappearance. The nucleolus, a rounded structure in the nucleus, disappears, right? So something disappears. Why? Because the nuclear envelope is broken. All right, so mitotic spindle development. The cell's two centrosomes move to opposite poles. Opposite poles of the cell, and microtubules assemble between them to form the mitotic spindle. Prophase occurs after interphase when DNA has already been replicated. So by the time it's prophase, the DNA has already been replicated, right? So the cell's two centrosomes move to opposite poles. They're going in different poles, and microtubules assemble between them. So what you're actually doing is you're organizing everything in different locations and having like a little bit of framework. So let's say if you want to study for MCAT, you're doing is biochemistry on one side, you're putting physics on one side, you're putting biology on one side, trying to organize everything, putting framework. So that's where the microtubules come in. Because why? Microtubules are cytoskeleton. They provide framework, right? Imagine having no framework and you just have a cell. Everything will be gooey, right? So that's what it is. All right, so these are your spindle fibers. That's the centromere, right? This little blue thingy is a centromere. Non-sister chromatids. So these are non-sister chromatids. 1, 2, non-sister. This is nuclear envelope, which breaks down. As you can see, it's breaking down. These are sister chromatids. All right, so these two together, sister, but the green and the blue one is not the sister. This is an X-shaped chromosome. It looks like an X, right? Metaphase is a stage of mitosis or cell division when chromosomes align in the middle of a cell, right? So metaphase is in the middle. That's why it's middle, right? Chromosome alignment. Whenever you think about metaphase, think about Facebook. Think about meta. It's in the middle, right? Chromosomes alignment. Chromosomes condense and move to the center of the cell, where they align along the metaphase plate, also known as the spindle equator, right? So it's in the middle. So it's in the equator. Kinetochore, microtubules. Microtubules extend from opposite ends of the cell and attach to the kinetochores around the centromere of each chromosome, right? The microtubules then pull the sister chromatids back and forth until they align. So, you know how sometimes, we have to use a pulley to align things. We can't do it with our hands, but then we have to use some stick or anything like that. That's exactly what it is. So, kinetochores are joined to the centromeres only to make sure they are, the sister chromatids are aligned in the perfect location. So, you're using something to attach it to something and then slowly prepare for the anaphase, right? Metaphase checkpoint, the cell ensures that chromosomes are aligned and the kinetochores are attached before entering anaphase, the next phase of mitosis. The metaphase is the checkpoint, right? So it's very important that during metaphase, you're in the middle, we might as well just check it if it's right or wrong, right? Alright, so this is the metaphase, this is the plate, right? As you can see, they are all aligned in the middle. Anaphase. Anaphase is the fourth phase of mitosis, the process that separates duplicated genetic material. So now you understand why the checkpoint is in metaphase. Why? Because anaphase is after metaphase, right? And this is where the duplicated genetic material is going to be separated into identical daughter cells. So if they're not perfect, if they're not identical, there's something wrong with it. We can stop it. That's exactly why you have a checkpoint. During anaphase, sister chromatids separate and move to opposite poles of the cell. Why? This is where, what you actually did was separated. So biochemistry should not mix with biology. Physics should not mix with cars. They are all in different locations, right? All right, separation. The enzymatic breakdown of cohesin, which link the sister chromatids together during prophase, causes separation to occur. So separation, the enzymatic breakdown of cohesin, right? Cohesin is cohesive, it sticks together. But what are they doing? They're breaking down cohesin, which linked the sister chromatids together during prophase, causing separation to occur. So if we want to separate something, what we have to do, we have to make sure the glue comes off of it, right? So that's what happened. Movement, the mitotic spindle made of microtubules pulls the separated chromosome to opposite poles of the cell, right? So mitotic spindle made of microtubules pulls the separated chromosomes to the opposite poles. They are opposite poles. Subphases. Anaphase can be divided into two subphases, anaphase A and anaphase B. In anaphase A, kinetochore microtubules shorten and draw the chromosomes toward the spindle poles. In anaphase B, astral microtubules pull the poles further apart and interpolar microtubules slide past each other, right? So in anaphase A, kinetochore microtubules shorten and draw the chromosome towards the spindle pores, right? So they're shortening it and then drawing centromeres to the spindle poles. In anaphase B, astral microtubules pull the poles further apart and interpolar microtubules slide past each other. So interpolar microtubules are sliding past each other, but astral microtubules pull the poles further apart, right? To me, this whole thing looks like science fiction. It's like you're in a Star Wars movie, right? Anaphase, right? So this is sister chromatids, right? These are sister chromatids. As you can see, these are blue. So this and this are non-sister, but this and this are sister, right? So as you can see, these ones are sliding each other, interpolar, right? The cleavage furrow, this is like a starting of two daughter cells. These are centrioles. These are kinetochore microtubule. That's plasma membrane. As you can see, the first time we see the formation of plasma membrane. Telophase in mitosis is the final stage of cell division where the separated chromosomes reach the poles of the cell. A new nuclear membrane forms around each set of chromosomes and chromosomes begin to decondense, essentially reversing changes that occur during prophase, resulting in two distinct daughter nuclei within the cell, ready for the cytoplasm to divide in cytokinesis. It is the last phase of mitosis before the cell splits into two separate daughter cells with identical genetic material. Function is to re-establish nuclear membrane around each set of separated sister chromatids, making the end of nuclear division in mitosis. So what happens during telophase? Chromosomes reach the opposite poles of the cell. The mitotic spindle disassembles, right? A new nuclear membrane forms around each set of chromosomes. The chromosomes become decondensed and return to the interphase appearance. What follows telophase is cytokinesis, where the cytoplasm of the cell divides, creating two separate daughter cells. So the telophase of mitosis, the nuclear envelope reappears. As you can see, this is the nuclear envelope and it reappears. Cytoplasm starts to divide. So this is the cytoplasm and they start to divide. Cytokinesis is the final cell stage, a stage of cell division where the cytoplasm of cells physically splits into two separate daughter cells, occurring after the nuclear division process of mitosis is complete, essentially. Cytokinesis is the splitting of cytoplasm that happens following mitosis, resulting in two distinct cells from one parent cell. Key point about cytokinesis is mitosis focuses on nuclear division. During mitosis, the genetic material chromosomes are replicated and separated into two identical cells, preparing the cell for division. Cytokinesis is the physical separation. After mitosis, cytokinesis is not part of mitosis, it happens after. Cytokinesis occurs where the cell membrane pinches inward, dividing the cytoplasm and organelles into two distinct daughter cells. Cytokinesis usually begins during the latest stages of mitosis, specifically in anaphase, telophase, depending on the cell type. Okay, different mechanism in plant and animal cells. In animal cells, cytokinase involves a cleavage furrow formed by contractile ring of proteins, while in plant cells, a cell plate is formed between two daughter nuclei to create a new cell wall. Cytokinesis of mitosis, right? So cytokinesis usually occurs after telophase, right? It is not part of mitosis. Nucleus reappears. As you can see, the nucleus appears, and these are two daughter cells that are formed, right? Alright, so mitosis and equational division. So from one cell, what did you have? You have two. We have two chromosomes. Here in metaphase, you see an alignment, right? In anaphase, you see division. As you can see, these two sister chromatids are divided. And telophase, you'll see two. You'll see one here and one here, right? Alright, so cytokinesis, this is animal cell. This is plant cell. In plant cell, as you can see, there's a plate. Whereas here, it shows constriction due to a protein ring. And now you have two daughter cells. The furrow meets forming two daughter cells. So this is under microscope. This is prophase. This is anaphase. As you can see, they're separated. They're moving apart. This is metaphase, they're in the middle. So prophase, metaphase, anaphase, and telophase, right? So this is telophase. As you can see, the two daughter cells are created, right? So what happens when cell division goes wrong? Some cells complete cell cycle within 24 hours, while others may take years before going through the process of cell division. Your body has many different signals that control how much and how often your cells divide. When cell division goes wrong, the cell division may not always go on as it's normal. Some cells may divide too fast, while others may divide too slowly or not at all. When cell divides faster than they should, cancer may occur, right? So if the cell's dividing faster than they should, then the cancer may occur. Normal cells, this is the division of normal cell, right? Many cells that continue to grow and divide. Variations in sizes and shapes of the cell. This is cancerous cell. So nucleus that is larger and darker than normal. As you can see, it has a nucleus. It has a presentation of large nucleus. Abnormal number of chromosomes arranged in disorganized fashion, right? So something's wrong with the anaphase here. And the nucleus is too large. Cluster of cells without boundary. So there is no boundary here. Normal cell division is controlled process that replaces worn out cells, while cancer division is uncontrolled and can lead to tumors. Here are some differences between the two. Signals: normal cells only divide when they receive the right signals, but cancer cells divide even without signals and ignore signals to stop dividing. Right? Normal cells stop growing when they touch other cells, but cancer cells continue to grow and divide, forming tumors. This is exactly why sometimes, due to malignant tumors, we actually see a very high density of mass. It will be a very big lump. Yeah. Normal cells don't move around the body, but cancer cells can spread to other parts of the body through blood or lymphatic system, right? Lympha, the water, the body of water, right? Immune system, normal cells are eliminated by immune system, but cancer cells can evade the immune system or trick into helping them grow. So cancerous cells are tricksters. All they want to do is divide, divide, divide. They don't care about space. They don't care about quality. They don't care about any kind of chromosomal locations. They just want to divide. They don't care. Okay. Normal cells have 46 chromosomes, but cancer cells can have too many or too few, or the chromosomes can be reshuffled, right? So location is a concern. Normal cell division versus cancer cell division. Cancer cell use different nutrients and pathways to transform nutrients into energy than normal cells. Cancer cells can induce new blood vessels to grow toward tumors, which supply the tumors with nutrients and oxygen. Normal cells have a set function, but cancer cells have no useful function. Alright, so that's it for mitosis. If you have any questions, you can comment. Bye.